Indivumed is a global oncology company offering specialized products and services in the field of translational and preclinical R&D projects, as well as development partnerships through different business units. In addition to services in the field of IHC, proteomics and genomics, we collect biospecimens under stringent SOPs and hold a unique biobank. With the help of these resources, we support our customers in biomarker and target discovery, drug development and more.
As Indivumed business unit, we provide a wide range of ISO-certified and GCP-compliant services based on our own biobank and state-of-the-art laboratories. These services include, for example, IHC assay development, both fluorescent and chromogenic, in-house digital pathology, and other lab services. Further, our broad portfolio offers research services of various kinds, including immunophenotyping of tumors, drug testing in viable tissue cultures, spatial transcriptomics, and other methods in the field of proteome and genome analysis.
Hi, I'm Malik Khenkhar. I'm Senior Director IHC Development at Indivumed, and in my role I'm heading the IHC Development team at Indivumed, a group of dedicated scientists and technicians, and we develop IHC assays on automated staining platforms for our biotech and pharma customers.
Hello, my name is Daniel Billius. In my role as a senior scientist and deputy head of the IHC Development team, I oversee the introduction of new multiplex IHC technologies at Indivumed.
My focus is to lead and provide scientific advice to the team on various customer and internal projects, planning resources and tracking the achievement of our goals.
I mainly focus on the development of new multiplex IHC technologies in order to offer them to our customers, and I'm providing technical advice to our team.
My main area of interest is immuno-oncology, an area that I believe promises great advances in cancer therapy.
Next to immuno-oncology, my main interests are new multiplex in situ technologies, as these offer new opportunities to characterize the tumor microenvironment, for example in terms of the spatial relationships between different cell types and the tumor itself.
We wanted to implement the latest multiplex IHC technologies on a fully automated staining platform to meet the needs of our customers for their preclinical and clinical programs. The field of immuno-oncology is increasingly moving into this direction in recent years, and the Leica Bond RX staining platform and the fluorescent multiplex technologies developed for it were the optimal solution to move in the same direction with our services. We provide automated chromogenic IHC and fluorescent multiplex IHC services for the preclinical and clinical programs of our customers. Our services include assay development and validation, meeting the standards of the EMA and FDA bioanalytical assay validation guidelines, as well as chromogenic IHC and multiplex IHC prevalence analyses and preclinical studies conducted in compliance with GCP, including histopathological evaluation by in-house pathologists or a dedicated digital pathology team.
The main reason for us to choose the Bond RX platform was the multiplex technologies, and the platform itself were best suited for our needs, meaning they can automatically stain 2 to 5 targets in parallel with high sensitivity and maximum reproducibility. The biggest benefit for us switching to the Leica Bond RX was to be able to perform automated fluorescence multiplex IHC, in which up to 5 targets are detected in parallel, and in equally high quality we previously performed chromogenic IHC. The biggest hurdle we had to overcome when setting up automated fluorescence multiplex IHC was to elaborate a suitable set of experiments for multiplex IHC assay development and validation to establish the best possible protocols. For example, experiments to determine the optimal staining sequence of targets with different epitope stabilities, or experiments to determine potential epitope blocking effects between different targets on the same cell.
We run Akoya TSA-based and UltiView multiplex IHC on the Leica Bond RX because both technologies were developed on this platform, making it the optimal choice for these types of assays. The ability to also perform automated RNA-scope mRNA-ish and chromogenic IHC was another advantage of the Leica Bond RX.
The Leica Bond RX platform offers the highest possible flexibility. We can develop complete fluorescence multiplex IHC assays for any target molecule using Akoya TSA reagents and optimize each step of the protocol. At the same time, you can use valuable pre-developed assays such as RNA-scope, mRNA-ish, and UltiView multiplex IHC. The great advantage of the cover tile technology is that it keeps the forces of the tissue during automated staining, which could affect the tissue integrity, to an absolute minimum. Currently, about 20 people work with the system.
Yes, and training new staff is easy because the instrument is intuitive and easy to use. However, learning all the details of developing a multiplex IHC test including all the protocol steps that can be changed, takes some time due to the flexibility of the system, but it pays off.
One of our main requirements of a customized staining platform is a high degree of flexibility in creating protocols that allow us the testing of different staining conditions, step sequences, and using a broad variety of reagents, while ensuring a high reproducibility.
In the future, I can imagine that certain multiplex IHC tests will be performed for diagnosis and patient stratification for therapies in routine clinical practice to evaluate well-defined immune targets and cell types that can only be detected in the multiplex setting. Anyone not yet convinced by fluorescent multiplex IHC should best get involved with the technology as soon as possible. Everything is developing very fast, and the better the technologies become and the more useful data they provide, the faster and more widespread they will find their way into routine diagnostics.
My advice for colleagues adopting fluorescent multiplex IHC is to make sure you control for everything. Pay attention to the details and set up a good development and validation process, so you really know what you are working with. And of course, ideally, you have pathologists and digital image analysis specialists at hand to help you interpret all the data, which are also a very important part of working with multiplex IHC assays here at Indivumed.